Guten Tag. It's the extent of my German. Yes, I am Derek from Brown University in the computer science department. I'm a PhD candidate there. And I'm going to be presenting work with Soren Estrel, my advisor. Haplotype assembly, where we extend the normal diploid case to polyploid genomes, and also extend it to more than one genotype at a time. We're going to assemble people that have a tract shared identical by descent. And there's also some diploid haplotype assembly results that are new as well. Okay, so genome assembly algorithms produce consensus sequences, and some of the good ones can give you variants, or you can map reads back and infer the variants yourself afterwards. But the phase of these variants is unknown. So for two variants here, we have two different explanations for those variants, which of the alleles actually traveled together on the same haplotype or the same chromosome. But haplotypes are very useful for a number of bioinformatics applications. On the top left here, we have any type of pedigree inference or ancestral recombination graph reconstruction requires or is enhanced by knowing haplotypes. Just to the right of that, we have LD computation and certain statistics that require the knowledge of haplotypes. And on the bottom, here's another example. This is genomic imputation, where you have a sample that you're interested to impute unknown alleles for. And for this, you need a set of reference haplotypes, and you look for haplotype sharing to impute those missing alleles. So how can we do this haplotype assembly? How can we infer phase from sequence reads? Well, we can use the assumption that sequence reads are sampled from haploid fragments. So in this case, we have 3 sequence reads, and they're sampled from one haplotype or the other one. And what we're going to do is we're going to build these sequence reads similar to genome assembly, where you can imagine that these two reads overlap at the C allele, and we can build the larger AC-T haplotype by combining the two. So similar intuition. Informally, we can describe the haplotype assembly problem as given a set of aligned sequence reads and a set of variant calls. We want to compute 2 haplotypes for each chromosome or each scaffold. So very simply put. Okay, so the input is a set of aligned sequence reads. Here's a small example. Now we convert the alleles that are in the sequence reads to 0s and 1s. So 0 is the reference allele, 1 is the alternate. And if we had no sequencing errors, then we would have a very easy problem. We could just look at the fragments, and where they share a variant and differ in allele, they conflict. And this would represent 2 fragments that belong on 2 different haplotypes. So in this case, we have 3 fragments, and they all share both variants, and they all conflict in at least one spot. So we can make a graph where we have vertices for fragments and edges between 2 fragments if they conflict. Now, because there are sequencing reads, we have to have some sort of optimization, and there are different criteria. We can remove the minimum number of fragments, the minimum number of variants, or we can correct the minimum number of errors, and this is what's used mostly in practice. So in this case, we corrected fragment 3, the second variant in fragment 3 from a 1 to a 0. So there's been a considerable amount of algorithms in theory that have been developed for the haplotype assembly problem. Most of them work on this MEC model, so correcting the minimum number of errors in the reads. I'll be talking about today the HapCompass algorithm, which is our previous publication last year. And what we're going to do is step through the algorithm and the model, and we're going to show how we go from diploid to polyploid genomes. Okay. So the HapCompass algorithm is built on the fundamental data structure of the HapCompass graph or the compass graph. Vertices are variants, and edges can connect variants that are spanned by at least one of the reads. We annotate the edges with the phasing between those 2 variants. So this is best shown through example. Here's an example compass graph. We have 4 variants, and on the edges we have the phase. So I'm not showing the reads here, but you can imagine if you have more cis haplotypes than trans haplotypes, then you'd probably prefer this phasing. So there are a number of ways to phase it, but majority rule is just one of them. So the first concept in these compass graphs is edge decidability. So in the diploid case, if we had a read, then the read either falls into this phasing category or this phasing category. It's very easy to figure out which one it falls into. However, in the triploid case, just as an example, these are two different explanations for two adjacent variants. If we have a read that's a 0,0 read, we wouldn't know which one to put it in because they both share this haplotype. So we no longer have this disjoint explanation condition. So we have a combinatorial algorithm to decide what phasing is the best between two variants. We also have a likelihood model where we want to compute the likelihood of a particular phasing for an edge, giving a model for sequencing errors in reads, and the set of reads that overlap those two variants. And we can compute this rather easily. The second concept here is phase extension. So here's a piece of that graph that I showed before, and we want to extend the phasing from the first edge to the second edge, and we simply just join on that shared vertex. So that's rather simple in the diploid case because every variant has a 1 and every variant has a 0 allele. Now we start running into trouble when we have cycles in the compass graph. So here's an example of a cycle in the compass graph. We construct this chain graph. So a chain graph, and I promise this is going to be the last graph data structure. The chain graph has a vertex for every two vertices for every edge. So for instance, this V1, V2 has two different haplotypes in its phasing, and we create two vertices. And then we just do the same phase extension to add the edges in between adjacent levels. So between V1 and V2, V2, V3, the 1,1 haplotype would be extended by the 1,1 haplotype, and we see that on the edge. So in this case, the two disjoint simple cycles correspond to the two haplotypes. So as long as we have disjoint simple cycles, we have two valid haplotypes. Here's the other cycle in the compass graph. We notice that we no longer have two disjoint cycles. We have one large cycle. So there's no there's no set of haplotypes that are consistent with this chain graph. This is what we call a conflicting cycle. So now in the polyploid extension of diploid haplotype assembly, we have the same problem. We need to extend haplotype phasings from edges to paths. It's pretty easy in this example to extend this bottom phasing, but this top it's not so easy. So we have two different ways to extend. Here's an example of a polyploid compass graph that we're going to use. It's very similar to the last compass graph, only we've added one extra haplotype to each edge. We'll take a look at this first cycle. So this is the polyploid chain graph. It's the same as the chain graph before, only instead of having that cycle edge, we removed it and just added a source in the sink. So now a valid haplotype is a path from one source to the corresponding sink. And what we're going to look for is we're going to look for k disjoint paths, no disjoint paths in this graph. So in general, the k disjoint paths problem is difficult to compute efficiently. But because we have a special structure here, it's called a trellis graph, we can do this very efficiently. And how we do this is we first, from each source node, we do a modified breadth first search. We annotate each node with whether or not it's reachable to its corresponding sink. Then at each level, we just compute a matching. Here's a matching for the first level and the second and third. If we have a valid matching at each level, then we have k disjoint paths. The solution may not be unique, but we know we have at least one set of k disjoint paths. So let's look at the other cycle. So here's the polyploid chain. We compute the annotations on the vertices like we did before. And we notice that in the first level, we do not have a valid matching. So if we go back to here, you can notice that T0 is not even reachable by S0, and similar for T1 and S1. Okay. So how do we deal with these errors? So we can find the errors by this k disjoint paths algorithm. To deal with them, we just remove an edge, and we remove the least likely edge depending on our model. In this case, we can remove this top edge if we just assume that that was the least likely edge, and then we can compute the phasing, because we can always compute a phasing of a path in cycles that give us the problems. In short, the cycle basis algorithm first builds the compass graph data structure. Then it computes a spanning tree cycle basis. So this is a very fast operation. We compute a maximum spanning tree, then for each non-tree edge we add it in, and then we determine what the simple cycle is after adding it in, and the set of those cycles forms a basis. We'll call those fundamental cycles. Then we repeat the following process until all cycles are non-conflicting. We choose a fundamental conflicting cycle, we remove the edge with the least evidence, and then we rebuild the cycle basis. Then we can compute a haplotype phasing that's consistent with the resolved compass graph that doesn't have any of these conflicting cycles anymore. So to start the results off in the polyploid case, we have 1,000 genomes NA12878 individual from the CEU population. We simulated a third chromosome and we added some reads, third chromosome 22, and then we tested the greedy method, which I didn't describe, but it's in the paper, versus the maximum likelihood method. And we see that maximum likelihood does better. So x-axis is the number of reads, y is the correctly phased haplotypes, the percentage of correctly phased. And with little amount of coverage, you could guess that they both do equally as good or equally as bad, depending on how you look at it, because they're both just guessing. But as you get more coverage, we're up to about 97, 98% correct. And this is about 300,000 reads here. So in the diploid case, we have theoretical results that we show in our paper. I'm not going to go through them here, but we basically show that the optimization that we use and a related optimization where we remove vertices instead of edges are both NP hard. And for the following slides, I'm going to use 3 metrics to describe the results. The first is the minimum error correction, which is related to the optimization. It's just the number of bit flips in the reads that we need to use in order to map them all back to the haplotype. So you want very few bit flips. In that case, your haplotypes are representative of your reads. And the Boolean fragment mapping is the number of reads that do not back to your haplotypes. And the center one is very similar. In all 3 cases, you want low counts. Low counts are better. So we took this individual, the phase 2 data, the high coverage data from 1,000 genomes, and we haplotype phased using these 4 algorithms. We see that we also describe an implementation of an MEC hapcompass algorithm, but MWare still does, this is the original algorithm for the diploid case, still does the best out of the 4 that were chosen. So Levy et al. 2007 is the algorithm that was used to assemble Venter's genome, and HapCut is a very reliable method by Bathna and Bonsall. We looked at some Broad chromosome 20 PacBio reads, so we wanted to see if the effects, the accuracy of the algorithms changed if you have PacBio. If you're looking for the minimum error correction, so the minimum number of errors, then hapcompass does the best. Levy et al. Actually does very well in these first 2 categories, but does very poorly in MEC. And this is because the way that the Levy et al. Algorithm works, it first places reads, so it's more concerned about placing reads than it is correcting individual errors within reads. We can talk more offline if you're interested. Lastly, I'm just going to give the intuition about haplotype assembly and identical by descent tracts. So we say that an identical haplotype tract passed down from a common ancestor is said to be identical by descent. And what's new to the haplotype assembly model is you're assembling now multiple individuals on one graph or simultaneously. So of course you gain the increased coverage on the shared haplotype, but what's more important is you have a larger pool of variants to phase. So the intuition here is if you have a homozygous region in black, or even just a SNP desert, you can put as many reads as you want that hit the flanking region, but if your insert size is too small, you'll never connect the 2 flanking regions. So this is the biggest problem with modern sequencing when you just do the short paired end libraries. With the insert sizes, you can produce tons of coverage, but the insert sizes are so low, and you end up not connecting these flanking regions. However, if you have a parent, you assemble them jointly with the child, then you can bridge those flanking regions with the parent, and then you can use the shared haplotype in order to get the assembly for the child through that region. So if you do this trio haplotype assembly, we show that, so this is the phased haplotype block size, and this is the number of phasings. If you assemble them together, then you get more haplotypes, and you also get larger haplotypes. Just to summarize, we showed in our paper that the two optimizations are NP-hard. This motivates the usage of our heuristics. We showed that identical by descent tracts enhance haplotype assembly, that you can model polyploid haplotype assembly. It's much more difficult, but we showed that the edge phasings in the compass graph are accurate, and the method is very fast. So all we're doing is computing spanning trees, we're computing disjoint paths and trellis graphs, and we're computing matches, and all of these can be done really fast. So the program is available from this website. If you're going to use the polyploid version of it, or even the regular version, you feel free to contact us for help or if you have any problems. Finally, I just want to acknowledge the organizers and the National Science Foundation for providing funding for me to travel here today. Our funding to do the research from NSF, 1,000 Genomes and Broad for the data, and my advisor, Soren Israel, who's given the peace sign here, for help with this research. Thank you.
Yeah, so if there's any questions, please come forward to the middle microphone as we're having a recording. And
thank you very much for the real nice talk. I was wondering how your algorithm's complexity and accuracy would scale as the ploid increases. Like instead of triploid, what if it's tetraploid?
Yeah. So we purposefully showed the triploid example because it still only has 2 explanations. As you start growing the ploidy, then you have a lot of explanations on the edges.
Is it exponential?
What's that?
Is it exponential in the ploidy?
I don't believe it's exponential. I don't know offhand, but I don't believe it's exponential. So if you have quadra or tetraploidy, I think the most that you have is 3 explanations on the edges. Mm-hmm. It does grow fast, but I don't believe it's exponential. I'll have to check.
Mm-hmm. And the online available tool right now can handle tetraploid and larger ploidies?
So right now, HapCompass just computes a assembly that's consistent with the compass graph. It's not necessarily the best assembly, so we have to work on getting the best assembly out, but it will work with, it should in theory work with a tetraploid genome.
I have one more quick question actually. As far as I understand, the algorithm only gets the information from pairs, right?
Yeah.
What if in the case a read overlaps multiple heterozygous SNPs? Does it use that kind of information, and how do you think it could be augmented to handle that?
So it uses that information indirectly. So if there's k SNPs in a read, then it has k choose 2 pairs, and it encodes all of that information as separate observations in the compass graph. And what happens with these cycles, if you have multiple cycles and they're all conflicting, what we do is we don't actually just select one cycle. We have a set cover formulation, and we select an edge that resolves the most number of conflicting cycles. So it's used indirectly.
And do you have a plan to convert this into a hypergraph problem for better resolution maybe? Because a read can splitting into pairs is not exactly phasing for that particular triple, right?
No, we preserve the phasing. We produce the maximum length phasing.
Okay, we can talk offline, I guess.
Sure, yeah, absolutely.
Hi, I just wonder whether your software can also be used by reads which are done by complete genomics.
In theory, there's nothing stopping the software from using any type of reads. So we showed that it worked well with PacBio reads, and that's dominated by indels. And also conventionally we use it with Illumina. I didn't show here, but we had a talk in HitSeq where we used Illumina, PacBio, and 454 all together, and it worked very well. So I would have to check the error model to see if anything funky happens, but in theory it should work with reads produced from.
Now we have a bit more time if there's any other questions. I was actually curious about the uptake of the method. I mean, it seems to have generated some interest here, but has there been any other uptake from other groups so far?
So right now we don't track people that download it, but we've had 3 or 4 people asking us for help with it. So it is written in Java, so it's very easy to use, and it's documented pretty well. But we're always open to help with questions, you can just email me or email Soren. Should be easy to use for any computer with a JVM.
Okay, so there's no more questions. Please let's thank Derek once more. And we're in the embarrassing position of running slightly early, so to allow the people coming from the next sessions, we